Okay, so here I want to talk about vaccine ontology. It has been here for many, many years, I think 18 years. I just put a few names here. So Jay and Anthony is here. Jay is, they are both working in my lab. And Anna Maria and Barry, they are my official consultants. And I have many more I will introduce later. So before I introduce more about vaccine ontology, I want to say, yeah, my lab has been also doing some other ontology work, like including faces, right? Like Bill and John. And that's one of the few behavior-related work that Bill Hogan has mentioned. So I'm a co-investigator for the project. I am also a co-investigator for a kidney precision medicine project. So it's a NIH-funded project. So this is more like a second phase or second five-year project. They said that they are going to fund, the NIH is going to, NIDDK is going to fund 3 phases. So we are still in the middle of the project. I'm more like the person taking care of the ontology. So how to go back? Okay, so yeah, we developed some 2 ontologies. One is ontology of precision medicine and investigation. We use OB as our parent ontology, and the kidney tissue atlas ontology is more application ontology. We also have been working with Alex and Bill and many other people working on some project called cellcast.org. So we developed a project several years ago. We're trying to apply for an NSF grant. We didn't get a grant, but still we are working on it. So we are using cell ontology and cell line ontology. And yeah, my primary appointment in Michigan is from the lab animal medicine. So I kind of finally found a way to collaborate with my other colleagues in my kind of unit. So I'm kind of working now on some lab animal model focused research. Okay, finally, let me come to the ontology. So I want to say, talk about a little bit of history. I would say like Barry, thank you, and also actually Lindsay Cowell. So I think you are the ones to bring me to the field. So I just checked. It's exactly about 18 years ago, like September 17 to 19. So there was the IDO meeting in Cold Spring Harbor. So that time we have 2-day meeting, September. And then in the end of the last day, I said, oh, my lab was developing the violin vaccine database. I think we should develop something called a vaccine ontology. So everyone said, oh yeah, go ahead. So then I got help. And then I checked my record, like April, the next year, April 2008. So Barry had a kind of a workshop, ontology workshop, just here in Buffalo. I think at that time you said, oh, this is probably will be your last one. So you said you will like record your video and later release to others. So I was the one actually in person. I feel very lucky. And then later, I think Barry, you introduced me to connect to the OBI, right? To Beyond Peter and Chris Stockard. Basically, I eventually got a letter from, I think Beyond, you wrote a letter for me. So I applied for the R1 grant. In the next year, 2009, I got a grant. It's my first R1 grant. With the grant, I was able to get my tenure actually eventually. So thank you all, yeah. So just a little bit of brief of introduction. And then we basically initially kind of was to use it to integrate data, right? To integrate the data with our vaccine database. And we be part of the OBO Foundry. And currently we have we have over 11,000 vaccines. So this is a paper we just submitted a couple of weeks ago or last month to the scientific data. And yeah, and we have a lot of people here, right? Like Alex, Bill, Peter, Beyond, and Barry. So this is the ontology. The overall structure, we use BFO as the high level, and we use OB, we use many other things. And then this is how we represent a typical vaccine, right? Like, okay, we often call that ontology design pattern. So how you represent a typical vaccine. Due to time concern, I'm not going for detail for now. And then this is the total number of vaccines we have produced in here. So you can see we have licensed vaccines, clinical trials. So clinical trials, I want to say, is like we want to actually, we have intention to represent every single vaccines, unique vaccines in clinicaltrial.gov. But that's a bigger goal, yeah, because they have thousands. So we just started, yeah. And then we have research. Initially, there was more like a research angle. So as long as you have lab animal models, you can verify the vaccine works in the lab animal model. We collect them. So we collect a lot of the vaccines using the lab animal model idea. And then applications, I just summarize, and then I'm going to give some simple examples. So we can initially standardize data from our violin database, and then we represent the knowledge and represent the data, and then support different kind of studies. And then we also use it to support a lot of literature mining later. So just give some examples. So in here, we can see Beyond mentioned that HIPS, PC, I know IEDB also use VO, and import also use VO. I'm going to have one more slide. And then vaccine adjuvant companion, actually, I want to mention because they contact me rather than we contact them. They say, oh, they contact me like Wolfgang say, oh, I'm developing the vaccine adjuvant companion. It's an intramural project. So basically, NIH funded a lot of vaccine adjuvant projects. So if you funded, right, if you got some adjuvant build up, you have to record. So this is more like that internal regulatory system. Then he told me, hey, we want to have for every vaccine adjuvant, we want to have a vaccine ontology ID. So then actually, it was a very good collaboration. So we have added 200 or 300 vaccine adjuvants for them in vaccine ontology. Okay, I come back to the OMOP. I know Barry is very critical to this one. I want to say I have been working with OMOP for 3 years now. So they are very famous. So even before COVID, it's already famous. But in the COVID, right, they have over 22 million patient record in the N3C. They use OMOP CDM. And also there's another famous one. It's called All of Us, right? All of Us common data model. They also use OMOP common data model to represent them. So if you look at the common data model, I mean, they are not an ontology model. They are more like a relational database model. They only have 10 to 20 relational database classes, and then they represent all the things together. But they do use ontology. They don't develop their own ontology. They use existing ontology to synthesize their data. But then the vaccine part, actually, they had a lot of problem. I have one more slide. So for vaccine part, they use RS-norm to represent the vaccines, right? But RS-norm is very good at drug representation. And RS-norm is more like a centralized for individual drugs or vaccines. Here, the vaccine, generally speaking, is considered as a drug. So, but they don't have good hierarchy. So then they had a problem. And somehow I was able to get connected with them. Actually, I presented a work in 2018 in OMOP, like in DC meeting. And I think Dr. What's called the Ralph University, Andrew, what's the name? Yeah, I want to say, yeah, he actually recently passed away. I feel very sorry about that one. But actually, he came to my poster. He said, oh, great. And somehow he introduced me to the ODC community. So basically, I'm a co-chair now, actually, with ASIA. We kind of trying to use vaccine ontology, to represent the vaccines in the hierarchical structure. So yeah, so that's kind of thing. Next month, there will be another meeting in New Jersey. I will attend there to more like represent our vaccine ontology to hopefully, we are still not there yet. It's more like HTO, right? The human phenotype ontology. They want to become part of ODC, but still they haven't succeeded yet. And we have not successful yet either. Yeah. So we'll see. So I want to give one more use case for the import, right? So this is internally, right? Internally, you can download the data. So each study, you have a vaccine ontology ID. So if you don't have, so usually ask us, right? So each one has an ontology ID. So this is how they can represent them. But sometimes you don't know the value of it. So then we say, okay, why not let's get all the ontology, vaccine ontology IDs and do something, right? So we have our tool, of course, like OntoFocus, right? So basically, okay, we get the IDs, we run OntoFocus. So we use something called like a intermediate level thing, and then you can build up this one. So you can clearly see, oh yeah, so all the import, right, using this kind of ontologies, you know the structure of the vaccines that have been reported to import for their studies. And then we also developed something called VGET. So VGET is a project funded by UHO2. Actually, it's an import pilot grant. So I worked with Dr. Guanmin Wu from Oregon for 2 years, 3 years. So Guanmin Wu actually is the primary developer of the real-time pathway. So he's a very good developer. So eventually we developed this kind of interface, right? And then we have, this is a vaccine ontology thing. The left side is vaccine ontology thing. So then if you look deeper, right, it's more like two files. So one is a metadata file. So it has all the metadata for many studies. Another one is normalized gene expression data. So we actually were wondering that time, like, can we do something more to standardize data? And then in the kidney precision medicine, we had the same issue, how to standardize the kidney precision medicine data and the infectious disease as well. So actually, I was thinking, actually also partly inspired by OMOP. So OMOP common data model, right? They can represent the EHR data in a very nice way. So I'm thinking, okay, OMOP data does not represent experimental data. Can we represent experiment data using something similar or something we call the common data model? So I say, okay, what's the basic idea of representing such experiment data if we want to do it? So then basically I got the idea. So here we focus on like a process, right? So 3 processes. So study, experiment, assay, there are 3 processes. So one study can have 1 or more experiment, and one experiment can have 1 or more assays. So difference is experiment generate samples. The experiment from organism, and organism can have some intervention. So experiment generate samples, and sample is the input of assay. And among all these, you can generate different kind of experimental factors, so you can affect the result. And then we go further, right? So it's still starting experiment assay idea. Then you have organism and the intervention, like vaccine, immune response, infection, or surgery. Anything can be intervention, right? And then the occurrence is more like natural. It's not man-made. It's like you have some comorbidity. And then you have samples, and sample can be part of assay. So like import has more than 10 assays. But then we say, oh, just assay is enough. One assay can be enough. Again, this is not a ontology. This is more like a data model. But you can use ontology to standardize data. So we use this data model to generate the original database, right? So this is actually like Anthony's here. So it's his PhD thesis. So he basically, based on the model, he generated, so each of the tables, right? Each of these become a recent database table. So once you put all data in, so he's use VGET, right? You have two files, then you put them into the 10 tables. So you can say, okay, one standardized query, right? What are the genes stimulated by the vaccine in females at day 7 after vaccination. So you can see, okay, I can now say, okay, I join three tables, right? And then I say, I can kind of put the filtering. It's very easy actually. So you can get results. Yeah, I think due to time, and actually the paper also was submitted to the journal, so it's under review. So we have a lot of information. But we think this kind of idea is not only important anymore. It can be used for kidney, can be used for infection, can be used for, I would think, actually, I would recommend this one for more broader, yeah, just kind of the idea, right? Because otherwise you have import, why you need another one? So basically, this is more than import. Okay, so that's, I think, more like this one. So I want to summarize my work. So basically, I presented how vaccine ontology can be used to represent rich vaccine knowledge, over 11,000 vaccines now, and many applications. I give some applications, and I presented the OMO of CDN. It's very popular, but the vaccine part is not good. I feel like it's very good for us because before we came from more like vaccine research, and now we want to go for the EHR. It's a very good opportunity. I know still a lot of issues we're trying to solve with them. And I presented Anthony's work on the study experiment assay common data model. We see we can use it to represent the different experimental studies and integrate them together with a very simple neutral model. So what's next? From biology to behavior. Actually, Barry just gave one sentence, one comment, which actually inspired me a lot. So basically, you said, oh, a lot of biology study, right? But now behavior, phenotype, but the gap is huge. So I feel like the CCDN one is more like experimental side, and the almost CDN is more like a EHR side. So can we fill out the gap using this kind of a study? I don't know. Yeah, we can discuss this. Okay.